Our lecture today is about the liver. Objectives. First of all, we are going to describe the secretory functions of the liver and their control, describe the metabolic functions of the liver and their disorders, describe the compositions and the regulation of the bile secretions. This is a picture about the metabolic functions of the liver. First of all, we are going to discuss the carbohydrate metabolism. First of all, the storage of glycogen allows the liver to remove the excess glucose from the blood, store it, and then return to the blood. When the blood glucose concentration begins to fall too low, this is called the glucose buffer function of the liver. The second function is the conversion of the galactose and fructose to glucose, and the third function, we call it the gluconeogenesis. When the blood glucose falls below the normal, large amount of amino acids are converted to form the glucose. About the metabolic functions of the liver regarding the fat. First of all, bile salts produced by the liver are essential for digestion and absorption of fat. The fat-soluble vitamins stored in the liver, such as vitamin A and vitamin D. In addition, water-soluble vitamins such as vitamin B12 also stored in the liver. Fat is metabolized when required and it's converted to ketone bodies. This is this takes place when the person suffers from prolonged starvation. The liver also the major site for the synthesis of large quantities of cholesterol, phospholipids, and the liver is the site for conversion large amounts of carbohydrates and proteins and form the fat. Regarding the protein metabolism, the liver is the main organ for the deamination of amino acids with the production of ammonia, and the liver is the only organ that can convert the ammonia to urea. Therefore, in liver failure, amino acids in the blood will increase and appear in the urine, and the plasma ammonia concentration rises rapidly and result in hepatic coma and death. The ability to synthesize the certain amino acids and also synthesize the other important chemicals components, and then these chemical compounds are formed from amino acids. Regarding the blood, the liver plays very important role in the formation and destruction of red blood cells. It is the major site for the formation of red blood cells in the fetal life. It stored factors principal for the normal maturations of red blood cells such as the iron and cyanocobalamin اللي هو vitamin B12. Can remove from the blood the toxic materials اللي هو bilirubin that formed after red blood cells destructions or breaking down, and the excess or excrete the bilirubin down through the bile in the bile duct into the duodenum. And the liver also manufacture the plasma protein and blood clotting factors such as the prothrombin and fibrinogen. Regarding the poisonous, many normal physiological substances, also the drugs, are modified by the liver before begin to excreted by the kidneys, and this is called the detoxification. The excretion of bilirubin in the bile. Usually the bilirubin is the greenish yellowish pigment responsible for the golden yellow color of the bile, and it's formed as a major and usually a product, and it is the major product that take place after destruction of red blood cells and destruction of hemoglobin. Also it is fast soluble, therefore highly soluble in the cell membrane, and it is very toxic, have the ability to pass through the blood-brain barrier, and it is secreted in the bile, and it is one of the very important functions in the liver. طبعا هاي example to ما يسمى بالdetoxification function of the liver. الexample مالته انه the getting rid of toxic materials such as the bilirubin excreted as a bile pigment through the bile outside the body. Jaundice means yellowish tint to the body tissue, including the yellowness of the skin and also underlying mucous membrane and deep tissue. The usual cause of jaundice is large quantities of bilirubin in the extracellular fluid, and it is either conjugated or unconjugated bilirubin. The causes of jaundice: we have the excess production of bilirubin caused by excess red blood cell destruction as in hemolytic anemia, or inability of hepatocyte to conjugate free bilirubin as in case of liver disease, or obstruction of bile duct or the liver cells, preventing obstruction of bile ducts that prevent the excretion of conjugated bilirubin inside the bile duct. Bile secretion: first of all, the bile is required for the digestion and absorption of fat. The bile is formed by the liver epithelium اللي هي الهيباتوسايتس اللي هي يعني liver cells, and by the epithelial lining of the bile ducts. Usually the normal value about 250 to 1200 milliliter per day of the bile is formed and stored about 30 to 60 milliliter volume in the gallbladder. So the gallbladder is a temporary storage of bile. After being concentration as much as 5 to 18 fold by a continual absorption of water and sodium and chloride through the mucosa lining of the gallbladder by active transport of sodium through the gallbladder epithelium. إذن gallbladder تعتبر the temporary storage of liver of bile. العفو. البايل يعني ينصنع تقريبا 250 to 1200 مللتر، لكن inside the bile gallbladder عادة يكون كميته 30 إلى 60 مللتر because of the continual absorption of water and sodium and chloride through the mucosa of gallbladder. فيصير عندنا يعني concentration of the bile going to increase. Now the formation of bile. The bile constitutes is the bile cells, which is يعني essential for the يعني absorption and يعني digestion and absorption of fat, and it is about half of the total solutes of bile. عندنا bile pigments اللي هي البيلروبين والبيليفردين. We have cholesterol, we have phospholipids, we have fatty acids, we have water and electrolytes. The bile acids are released with the electrolytes sodium, potassium, and combined with the bile pigment together forming the bile salt. إذن the bile salt عبارة عن bile acids، عبارة عن electrolytes، عبارة عن bile pigments, and it drain into the right and left hepatic duct, then to the common bile duct, which join with the cystic duct forming the common bile duct اللي هي join the pancreatic duct opened in the ampulla of water اللي هي located in second part of the duodenum and surrounded by ما يسمى بالsphincter of Oddi. The bile formed continuously, but it is not continuously released to the second part of the duodenum. The bile stored and concentrated normally in gallbladder. إذا عرفنا بأنه gallbladder تعتبر temporary storage of the bile وليس لها أي علاقة بصنع البايل. البايل usually synthesized by the hepatocyte. The bile released to the duodenum is about 0.5 liter per day, and the bile salt consists of bile acid, sodium, potassium, and it is responsible for fat digestion. About 94% are reabsorbed by the active transport process in the distal ileum and enter to the portal blood circulation and pass to the liver. The small quantities of bile salts lost into the fecal materials are replaced by new amounts formed by the liver cells. The bile salts reabsorption occur in the distal ileum, and the removal of distal ileum either يعني ميجالوبلاستيك انيميا. ليش يصير عندنا ميجالوبلاستيك انيميا؟ لانه راح يصير عندنا يعني الابزوربشن اوف سيانوكوبال امين اللي هو فيتامين بي 12 باي ريسبتور ميديتد اندوسايتوسيس الموجود يعني عاده النورمال سايت فور ري ابزوربشن او يعني اوف ذا سيانوكوبال امين يكون بالديستال اليوم. لما نشيل الديستال اليوم ايش راح يصير عندنا؟ يصير عندنا ميجالوبلاستيك انيميا. another complication راح يصير عندنا بايل سالت اللي هو يعني usually help in the absorption of fatty acid and اللي هي مثلا الفاتي اسيد والمونوغليسرايدز والكوليسترول واذر lipids from the intestinal tract and يعني forming a complex with it نسميها الميزلز and it is water soluble and easy absorbed when distal ileum without the presence of fat يعني bile acid usually in the intestinal tract up to 40% of the lipid are lost in the stool. يعني إذا bile salt together with the fatty acid تسوي لنا ما يسمى بالميزلز to be easy to be absorbed by the intestine. إذا ما عندي bile salt راح يصير عندي lost of lipid in the stool and the fat are not absorbed equally. The fat soluble vitamins also not absorbed equally مثل فيتامين A فيتامين A و D و K و E كده تعتبر fat soluble vitamins usually not absorbed يعني not absorbed يعني good absorption but lost by the stool. Therefore it's an decrease reabsorption of fat or bile acid lead to decrease the fat digestion and absorption and lead to ما يسمى بالستاتوريا. ستاتوريا صار diarrhea but يعني fecal material تكون bulk offensive and pale. عادة كريزي. يعني شنو كريزي؟ يعني الفيكال ماتيريال تكون دهنية. ليش؟ لأنه ما عندي بايل سولت reabsorption وبالتالي راح الفات lost by the stool والستول يكون الملمس مالته كريزي ويكون bulk ويكون offensive and pale. Usually the bile deficient from enzymes and therefore the digestion of fat by the bile عن طريق ما يسمى بالإيمولسيفيكيشن. يعني احنا عادة البايل احنا قلنا it is essential for digestion and absorption of fat, different types of fat. لكن البايل deficient from enzymes, digestive enzymes. يعني شلون نسوي لنا digestion؟ عن طريق ما يسمى بالإيمولسيفيكيشن. عادة molecules of fat tend to clump into a fat globules and the bile salts affect the fat globules much like the soap. Reducing the surface tension and break the fat globules to small droplets and then enable the digestive enzymes to يعني break down this small fat globules and lead to يعني be easy to be digested and then absorption will be easy. إذا يعني عن طريق الإيمولسيفيكيشن يشتغل البايل كأنه مثل الصابون مثل المنظفات يقلل السيرفيس تنشن ويحطم لنا الفات جلوبيولز تو سمول دروبليتس حتى يساعد الدايجستيف انزيمز اللي تطلع لي من السمول انتستين ومن البنكرياس وبالتالي يعني وحتى يعني تسبب لنا destruction of this fat drop. Now the control of bile release عن طريق طبعا 95% controlled by the hormones اللي هي الكوليسيستوكاينين بانكريوزايمين that is released from the duodenal mucosa. وطبعا the major stimulus for cholecystokinin pancreozymin hormone العفو هو الفات present of fat in the chyme. The gallbladder contraction and sphincter of OD relaxation lead to bile that flow into the duodenum. And also the secretion hormone also very important to lead to contraction of gallbladder and relaxation of sphincter of OD and also release of bile. إذا عندنا الهرمونال كنترول 95% عن طريق الكوليسيستوكاينين بانكريوزايمين وعن طريق السكرتين هرمون play very important role for the contraction of gallbladder, relaxation of sphincter of OD and release of bile to the duodenum. Only 5% controlled by the vagus عن طريق parasympathetic stimulation. يطلع لنا الاستالكولين act on the mucosa lining of the duodenum and lead to release of cholecystokinin pancreozymin and lead to contraction of gallbladder and relaxation of sphincter of OD. إذا بالحالتين يعني حتى الفيغاس نيرف الاستالكو راح يشتغل على السيلز that is located in the duodenum mucosa and release cholecystokinin. إذا في الحالتين يعني الكوليستوكاينين play very important role for contraction of gallbladder. بس أول مرة هو راح يطلع عن طريق وجود الفات الموجود بالكايم، والشيء الآخر عن طريق indirectly by the vagal stimulation by acetylcholine. So the person اللي suffer from vagotomy لما نقطع الفيغاس نيرف لأي سبب من الأسباب it will never أو it does not suffer from the decrease of release of bile. ليش؟ عندي تقريبا 95% الجول بلادر contracted by cholecystokinin يعني هرمون، وبالتالي ما راح يأثر على البايل ريليز إذا أنا قطعت الفيغال نيرف. Now خلينا نقول يعني ملخص لمحاضرتنا انه metabolic function of liver include carbohydrate, protein, fat, blood, the poisonic modification and the bile formed by the liver epithelium اللي hepatocyte that required for digestion and absorption of fat. طبعا البايل deficient from digestive enzymes and digest fat عن طريق emulsification. Control of bile majorly by the hormones الكوليسيستوكاينين والسكرتين and indirectly by 5% عن طريق الفيغاس also يعني indirect release of the cholecystokinin and lead to the digestion of fat, release of bile that are very important for digestion of fat. We have some questions about our lecture and thank you.